On the quiz, a number of you showed some common misconceptions that I'd like to address with this video. And the first thing is this idea of what is chromatin, that's chromatin, versus chromosome, versus the idea of chromatid. So what's the difference between these three? Well, it goes back to trying to understand what exactly is happening in mitosis, what's happening in the cell itself. And when you look at the cell and you just look at just the nucleus of the cell, so if we have a cell here and we have the nucleus of the cell, inside this nucleus is the DNA. And as we've talked about over and over again, there are 23 pairs. All right, and those pairs consist of one DNA molecule from mom and one from dad, and these molecules all have the same genes on them. So this gene and these genes are the same. Maybe this one's eye color. From this space here to this space here, this is for eye color. And the same gene is found here on this piece of DNA. These pieces of DNA are called chromosomes. But while they're in the nucleus doing their job, they're unwound. And so we call it chromatin. So all this DNA, all these 23 pairs, you can't distinguish one from the other. They're molecules. They have a beginning, and we'll talk about that in the next unit after we're done with meiosis. And they have an end, okay? Each of them have a beginning and an end. The one from mom and the one from dad, all 23 different chromosomes. But when they're all unwound, they're so long, they're 3 meters long each, and they're all unwound in this tiny little space inside a cell, inside a nucleus, that all you see is this kind of a bunch of little strings inside the nucleus. We call that chromatin. Chromatin. We discussed this before, but I hope it's clear now that when the DNA is unwound, and it's usually found and is found in interphase. So when it's interphase, any along the cell cycle, so that's G1, S, or G2, you're going to find it unwound in this form called chromatin. Okay. So be clear on that. That's what chromatin is. If you want to describe it, it would be grainy. It would be stringy. That kind of thing. But it's all unwound, and it's being used. And we'll talk about how DNA is used and how it controls the cell. But when it's being used, it has to be unwound. Okay, so that's chromatin. So a chromosome, remember, is this molecule of DNA, and it's a molecule of DNA that has a shape. Usually they draw it like that. That's how we see it under the microscope, something that looks like that. Really, what we're talking about is, remember, is a double-stranded DNA molecule that looks like a double helix. That's what this is. But it's a chromosome's all wound up. A chromosome, you only see a chromosome when it's all wound up, but it is still a single molecule of DNA that is wound up. It's just, remember, a molecule of DNA can be 3 meters long, so it has to be controlled. In order to fit inside a nucleus, its size must be controlled. So it is wound up around proteins called histones, and these molecules are wound up pretty tightly, and that's when we see them as chromosomes. Now, a chromosome is one molecule of DNA with all kinds of genes on it. And so a molecule of DNA that has a gene for eye color, or a gene for, height, or a gene for skin color. And these genes can be found in specific places, and on that chromosome, if it's chromosome number 1, you're always going to find the genes in these specific places. And these genes on chromosome number 1, code for specific things. There are different variants. Well, this will be small a, small b, small c. And what happens is that you can find then when you go through synthesis, remember we make a copy of this chromosome and it's connected by a centromere, right? And this copy, of course, has exactly the same DNA. So you'll find A here, and B, and then C over here. So you'll find the genes for that the code for those particular proteins for eye color, pigment, or for skin color, whatever, or height, or whatever the character may be. You're going to find them on the chromosome exactly where they're supposed to be. Because they're exact copies. That's what happens in synthesis, during S phase. And so you get two exact copies of chromosome number 1. And both of these, the original one came from Dad. So you're going to get 2 copies of chromosomes, exactly the same that came from Dad. All right, but remember we start off with 23 pairs. So when we're looking at this, we had one from Mom. We had this one from Mom. Exactly, this is chromosome number 1, right? But chromosome number 1 that came from Mom. Now that DNA is going to be a little different. Maybe mom has lighter skin color, or maybe Mom has blue eyes, or maybe Mom has green eyes, or whatever. So her gene, might be a big A, and maybe she'll have a big B, little b like Dad. But then maybe the C will be big C. Who knows? There's all kinds of different variations, but that might be the one from Mom. And so when you make a copy, during S phase, you make a copy, you end up again with an exact copy of this chromosome number 1 that came from Mom. And with exactly, this is A, this is B, and then this is big C. And so this came from Mom, and of course when we're talking about A, B, big A or little a, we're talking about the sequence, the code. What does this code say that needs to be produced? What color pigment, what amount of hormone, or what have you? So during S phase, they're copied, so you end up with this, as what some of you described as this X-looking chromosome. Because of the centromere that connects them. But understand that this is a chromosome. That chromosome is a wound-up, 3-meter-long DNA double helix molecule that when it's copied, you end up with 2 of them connected by a centromere. When they're connected, when these 2 are connected by a centromere after S phase, we call this we call each of these a chromatid. Let me erase that and make sure so I don't want to split the word up here. So we call each of those a chromatid, okay? Now understand what we mean by chromatid is that this particular area here, this is one chromatid, this is another chromatid connected by a centromere. So a chromatid is one copy of the chromosome, right, of the pair of chromosomes that are connected by a centromere. Now remember, these chromatids during mitosis, the whole point of condensing the chromosome or condensing the DNA into what we call a chromosome, or winding up the DNA so that is to package it so that it can be sorted, and so that during mitosis they can be separated clearly. So you get one copy from Mom and one copy from Dad into each new cell. So the little spindle fibers come in, they start to contract, they separate those 2 during anaphase, so you end up with one chromosome from Dad that has little a, little b, little c going into one cell, and one chromosome from Mom that has big A, little b, and big C going into the other cell. So a chromatid is an exact copy of the chromosome that originally was there, that was copied during S phase, and that later and during anaphase was separated into each individual cell. So that's what a chromatid is. A chromosome is a copy of DNA that has genes on it that is passed on from mother to father, from mother to child, or from father to child. And it's a copy of DNA that has genes on it. And you have two of them. You have one from dad and one from mom. And then we see them clearly when they are condensed, or when they, or a better way for, and by condensed we mean, you know, it's coming together. And another way of thinking about it is that this long 3-meter long strand of DNA is wound around histones, like taking a big some yarn that's just spread out throughout your table and then winding it into a ball. That's what you're doing here, but instead of a ball, you have this kind of a little sausage-looking thing. And you have two of them connected by this centromere. They form, yes, they do form this kind of an X-looking shape, and each chromosome is different. It depends on where the histone, where the centromere is located on that chromosome. What kind of shape it takes, and some shaped more like a Y, like the Y chromosome, and some are shaped more like an X. But whatever, the shape isn't important as far as that's concerned. Well, it is important for later usage, but for now, for the purposes of this discussion, it's not that important. What is important is that you understand that this chromosome here is an exact copy of this chromosome here, so that when during anaphase they're separated, this one goes into this cell and this one goes into that cell. They are, you're sure to get one copy of dad's chromosome into each new cell. And the same thing's going to happen to this chromosome number 1. This one's going to go in one cell, and that one's going to go in the other cell. So you get one copy of mom's chromosome number 1 in each cell. That would not happen. This is one method of making sure that you get the exact same pair of chromosomes, one from dad and one from mom, of each of the 23 pairs. Remember, there are 23 pairs. Some of you say 46, but it's actually 23 pairs, right? This is one of the 23 pairs. This happens 23 times, or 46 times, because there's 23 pairs. So this is one chromosome. The pair is this number 1. If you were looking at the pairs, when we say 23 pairs, we're not talking about this pairing. We're not talking about these two chromatids. That's why we changed the word to chromatid, so that we can discuss this as a chromosome, and we say this is chromosome number 1. And these are two chromatids, so they're two copies of chromosome number 1 connected by a centromere. By using the word chromatid and then calling this whole thing a chromosome, we can keep continuously talking about this chromosome and this chromosome as being a pair. And then if you look at it, when we look at this chromosome here and this chromosome here as being a pair. Because once they're separated, they're no longer chromatids, they're chromosomes again. And they're going to go off in pairs. You're going to get two number 1s into each new cell. But you want to make sure you don't get two copies, for instance, these two, because you'll have a different cell. This will produce, let's say, this cell is this is the gene for blue eye color, and this is the gene for brown eye color. You don't want one cell producing the blue color and the other producing the brown color. You want both of them producing whatever color it is that you're supposed to be producing for your cells. You want each of your cells to be doing the same thing. You don't want some cells doing one thing and other cells doing another. You don't have an organism then. You have an imbalance in the system. So by making sure that we have the same genes in each cell, we make sure that all our trillion cells are all communicating well. That's not to say that any one cell can't make a mistake, but that's why we have our checkpoints. So when they're going through mitosis, if something goes wrong during S phase, or something goes wrong during metaphase of the mitotic of the M phase, right? During metaphase of M phase, if something goes wrong, then the cell dies, and we make sure that all our cells are in line, all our cells are working on off the same page in the blueprint. All right, I hope this helped.